Without taking much time, I would like to introduce our first speaker, Dr. Kee Tan. Hong is currently a postdoc at Wellcome Trust Sankar Institute. He graduated last year from National University of Singapore and is now conducting research on saturation genome editing of developmental disorder genes in Dr. Matthew Hurl's lab. His lab specializes in applying new genetic technologies towards improving the diagnosis of patients with rare genetic conditions. With that being said, I ask you all to give your full attention to Dr. Kee Tan and help me in welcoming him to the stage. Dr. Kee Tan.
Hi. So I'm going to talk about the saturation genome editing of a developmental disorder gene. So that is around 60% of the de novo variant has been identified in the known developmental disorder disease, which is missense SNV. Most of this variant actually has an unknown significance, so it's classified as a VUS. So it becomes a very important question to design a good assay to study the functional impact of all of these possible variants in the genomic locus. So some of the assays have been reported so far. I have listed some of them over here. So I will only focus on the third method known as saturation genome editing. So what is saturation genome editing? In short, actually, we just make use of the CRISPR-Cas9 technology to lock in all of the possible single nucleotide variants into the cells. And we are looking at the cell survival as a readout. So for the cells carrying a deleterious mutations, will stop proliferating or dying at the end. And we only assay for the survived cells which carry the tolerated mutations. In our assays, with our first assay, we are focusing on the developmental genes called DDX3X. DDX3X actually is the RNA helicase which locates on the chromosome X. 1 to 3% of the female patients with unexplained intellectual disability may have the mutations in these genes. So far, the associations of the DDX3X mutation with the clinical outcome is less understood. So it is pretty difficult to assign the benign or the pathogenic variants on these particular genes. So, and also the DDX3X is recurrently mutated in several cancers, which leads us to the reasons why we want to study in these genes into more detail. So DDX3X actually have two major domains, the helicase ATP binding domain and the helicase C-terminal domain. In our pilot study, we only focus on the exon 14, which is located in the helicase C-terminal domain. In the exon 14, there is quite a number of the known CRIN-VAL variants have been reported to be pathogenic, so which can serve as a very good control for our assays. And there is also two nutmeg variants found on exon 14, which serve as a very good negative control for our assays. We expect that the pathogenic variant will be depleted in our assay. So this is our experiment design. We have designed two of the GAT1 and GAT2, which targeting the exon 14. And we have our HDR library, which consists of 500 sequences and have included all of the SNV and also some of the in-frame deletions. The cell line that we use is a HEP cells called HEP1 cells with the LIG4 lockout and Cas9 overexpressions. We found that with a LIG4 lockout and Cas9 overexpressions could enhance the HDR efficiency a lot. And we are harvesting our transfected cells at day 4 and day 11. We use the day 4 as an earlier time point and day 11 as a late time point for our analysis. And our library subsequently subject to MiSeq, and then we analyze the read count data. So we have actually optimized our conditions such that we can achieve 80% of the transfection efficiency, and which lead to relatively high HDR efficiency, which is about 50%. So that's why we can get a relatively good results from the assay. As I said, we have two guides. So the results from both of the guides that we have are quite consistent. So we have decided to merge these two data into one for further analysis. And here I show you some of our results. So we observe the stop gain variance is all depleted in our assay, so which is totally makes sense and also suggests that our assay actually is working, and the synonymous variant has almost a zero-fold change. In the missense variant, actually we observe most of them is depleted, but at the same time we also observe that some proportions of the missense variant actually significantly enhanced and rich in our assays. For the in-frame deletions, actually all of the in-frame deletions is almost like depleted in our assays, and only like a few of them is enriched, and only one of them actually has no functional impact. This is a big surprise to us. And for the intronic variant, there is only a few of them is depleted. So for the intronic variant, we have looked into a bit more detail, and we found that most of this intronic variant, which is depleted in our assays, is locating at the splice junctions, so as shown over here. And quite interestingly, that is one of the mutations over here is known as the, is found in the Greenbaum data and is assigned as the VUS. And over here, we found that this VUS actually has the same, behaves similarly to the loss of functions mutations, which has the same depletion as the nonsense mutations over here. For the missense mutations, we found that two of these nutmeg mutations actually has no functional impact, which is expected. And one of the clinical pathogenic variant actually has no functional impact as well. As what I mentioned before, it is quite hard to assign benign and also pathogenic variants for these genes. It is possible that this non-pathogenic variant actually is behaved like benign. And more surprisingly, we also found that there is two clusters of the missense variants. One of them actually is enriched and one of them is depleted. So for the enriched variant, there are 4 of them actually is assigned like the clinical pathogenic variants. So we think these enriched variants actually have already altered the protein functions, but it's definitely not the loss of functions variants. When we organize all the data into one single table, we could generate this kind of lookup table. So over here, we have integrated the in-frame mutation data and also all the amino acid substitution data into this lookup table. In future, we may want to generate this kind of lookup table for most of the development disorder genes, because we believe that this kind of lookup table will be very useful for diagnostic purpose. So over here, I would like to highlight the P513 residue. In this residue, actually is quite interesting because we observe that that is a nutmeg variant shown over here, which we found has no functional impact, and that is a few of the amino acid substitutions is depleted in the experiment, but the clinical pathogenic variant is enriched in these assays. Actually, which means this pathogenic variant actually could alter the protein functions, but is totally different from the loss of functions variants. So in summary, actually, I would just like to claim that the SGE that we have actually is successful, even though we performed this particular assay in the HEP1 cells. And also the data suggests that the single base mutations can result in either no functional impact or loss of functions or the altered functions on the DDX3X. And also all the reported clinical variants are not necessary to be loss of functions. Recently, that is a paper published in bioRxiv. In this paper, they studied the patients with a missense mutations in DDX3X, and they found that if the patient have a missense mutation in DDX3X, they are more likely to develop the polymicrogyria or the PMG in their brain imaging. This is what they observed, which means that some of the small folding in their brain imaging. But this particular PMG is not observed in the patients. They have the loss of function mutation, like the nonsense or the frame shift mutations. This strongly suggests that some of the missense mutations are not the same as the loss of function mutations. This is similar to what we observe in our data. So actually, this is work in progress. We have already initiated the work to study all the exons, all the 17 exons in the DDX3X. So in near future, we will complete the project and we will have the lookup table for these particular proteins. And we will going to validate the identified variants in the neural cells. As I mentioned, HAP1 cells is not a neural cells. So at the end, we would like to validate the variant that we identified in the neural cells. And in our department, we have initiated the study. We have extended this SGE study to as many genes as possible. At the first phase, actually, we are focused on around 100 of the developmental disorder genes. Yeah. Thanks.